Good evening, class. My name is Mrs. Ego Flora Okeke. I'm speaking on anatomy and physiology, HCM 112. In our previous class, I stopped at muscular contractions. That's muscle construction. Today we are going to study cellular organelles and the structures. Cellular organelles and the structures. Muscle cells contain organelles found in all cell. Muscle cells contain organelles found in all cells, including nucleus, endoplasmic reticulum, and the mitochondria and the Golgi apparatus. The amount of organization of cellular and the structure is highly different in muscle cells. Actin is found inside every cell in the body. This actin also helps in classes of food in specifying it according to the species, like protein, carbohydrate. But actin is special organized in the atmosphere of the muscle cell. Muscle cells also have extremely high number of mitochondria to produce APT for force generator. That is for force generator, which concerns the muscle. Recall that APT molecules is required for one motion to perform one power strokes. Look at the diametrical aspects. You see microcephaly, A brand, I brand, Z line, nucleus, and so on and so forth. Organelles and the structures specific to muscular cells. Organelles and structures specific to muscle cells. When you look at the diagram, you see muscle, you see the fiber, as I said before, see the peripheral nuclei, see the multifier, which is surrounded by the nuclear area, sarcomacus is surrounded by the nuclear area. Cross-section of the muscular cells. Cross-section of the muscle cells. The image is a light of microscopic image of skeletal muscle in cross-section, showing a group of muscle, that is muscle cells, is surrounded by epidermis and the group of surrounded by periphery. Blood vessels are seen passing through the image in cross-section of the site. It's talking about how blood circulates, that is blood sector in the muscle, and the fiber, how fiber works. You know, fiber is very good, helps in digestion. The magnified image showed a cross-section of all single-cell fiber at higher magnification with many modifier visible small dark circle. He's talking about magnifier. That is a magnification example, just like if you see a mirror, if you want to magnify in terms of microscopic, remember that is microscopic, that is from microscope, you see microscopic, so that the microscopic will be able to detect if there is anything in skeletal blood cell, in your skeleton, in your bone marrow, in whatsoever, it will be able to detect. That is magnification. It will magnify and detect any fault. Each skeletal fiber is a single cell produced from the fusion of many processor cells. Therefore, these four cells are therefore functionally quite large, with diameter of up to 100 micrometers. It's talking about the diameter and the length of up to 30 centimeters. It's talking about the diameter, the length and the diameter. Compare with the size of the skin, which is a cube of 20 micrometer in nearly every diameter. In my muscle fiber, sarcosa are run into the parent structure called the microfiber. So, both Z-disc and M-line hold microfermentation microfender in place to maintain the structural arrangement and the line. It helps to maintain the structural arrangement and arrange it in a sequential order. Microfiber also connected to each what? Each house by what? By intermediate, or the smell is in form of intermediate. It's the one that will help to separate this market and rearrange the body system. That's what this place is talking about. You know, that's why our body is a system. Something need to arrange it. This is some of special nomenclature associated with this fuel cell. Each skeletal muscle fiber cell has more than one muscle, have more than one numericals, which is called multi-numericity. The plasma member in this fuse select skeletal muscle cell is called what? It's called sarcocoelom. Because of what? Because it has its compartment, has its walls and functions different from one another. Remember that is concerning the fiber that will help things to digest, like help things to absorb. Another one we are going to see is that when you look at it, see also the sarcocoelom contain many structures similar to the plasma members of the cell. Plasma members of the cell. It also possesses structure unique to muscle cell. Unique, complete. The cellulina has transversal tubular or T-tabular, that is in form of tabular form, which is different things comes together in order to perform one specific task, which is indeterminate of sarcoma into the interior of the cells along the length of the microcell. It shows how it is arranged, the work one after the other and the sequentially, both slow, the one that will delay, the one that will be faster, and the one that will not be faster. The one that will show sequentially. Look at the diametrical aspect. Diametrical aspect, that is diagram. When you look at this diagram, you see different things there. You see even muscle, you see the muscle cell, you see the one that surrounding, you see the fiber. When you look at it, you see how it works sequentially. See how it works. That is the very, very significant, and how it goes. When you look at it, you see that blood vessel. Look at it down here, you see blood vessel. Look at this place, you see muscular fiber. Look at this place, see endoplasma between fiber. When you look at it, you see this place, you see bone. Look at this place, you see the other one. There are so many, so on and so forth. Muscle tissues. Examples of the muscle tissue. See cardiac muscle. Look at the cardiac muscle. Smooth muscle. Look at the smooth muscle. Skeletal muscle. Look at the skeletal muscle. In that skeletal muscle, look at human diagram. Look at the cardiac muscle cell directly to the heart. At times, people will say somebody have cardiac attack from the chest. It may at times mainly lead to death. Look at this one, skeletal muscle cells and the smooth muscle cells. And the smooth muscle cell. See this one, you see types of muscles. You see types of muscles. You see different types. Look at it diagrammatically. See the one that is called action posterior reaches terminal. Look at this one, atom, and so on and so forth. When you look at it, you see the other one, sense of organs. Sense of organs. Sense of organs. Senses provide information about the body and its environment. Humans have 5 special senses. 5 special senses: olfaction, brachial smell, gustion, taste, equilibrium, balance, and the body position, visible and hearing. Additionally, we possess general senses, also called somata sensation, which produce the stimuli from stimuli. When you hear stimuli, what you comes into your mind is what? Feelings in form of stimulus response, like temperature, pain, pressure, and the vibration. Stimuli, vibration. And look at some examples. Look at taste and smell. Taste and smell. Taste, also called gustation, and the smell, also called olfaction, are the most interconnected senses in that both involve molecules. This is talking about taste. In this respect, when you taste food, example, when you eat sour food, you'll be able to dictate whether that food is good or not. Smell, when you perceive the odor that is offensive, you may be able to dictate that that odor is not good. That is what? Offensive, that is not good. In the aspect of taste, when you taste something that is salty, you'll be able to know that it's salty. Look at different tests. Different tasting food have different attributes. Different tasting foods have different attributes, both helpful and harmful. As I said before, like you taste food that is soured, you'll be able to know that that food is sour, that is not even good. If you taste food that is raw, you know that is raw. If you taste food that is diet, you know that is diet. You may be able to dictate whether something is sweet in tasting. You'll be able to dictate that. Even when you eat spoiled food, you also be able to dictate. Another one is taste and odors. Taste and odors. Both taste and odor, stimuli, and are molecules taken in form of environment. Stimuli in form of environment. The primary taste dictated by human are sweet, sour, bitter, salty, and the mild. Be able to. The first 4 tests need little explanation. The first 4 tests need little explanation. Yes, a test. Dictating the test. They need 4 different explanation, like the sweet, the sour, the bitter, and the salty. The sweet, the sour, the bitter, and also salty. When you see something that is sweet, like sweet, you know that that thing is sugary content. The sour, when you eat something that is not even good, like soured soup, you know that that thing is very dangerous to your health. When you eat bitter something, you taste it. Example, when you put flagyl in your mouth, you know that it's bitter. Salty, when you cook food and know that it's salty, you know that it's salty, that is not even good. And also Another one is this: all others that we perceive are molecules in the air we breathe. Molecules. All others, they are the one that is offensive, they are the one that is not offensive. If a substance does not release molecules into the air from its surface, it has no smell. It has no smell. Example, perfume. When it spread perfume, you see that that perfume must show that something spread. If not, have no smell, it will be ordinary air. And if a human or an animal does not have a receptor that recognizes a specific molecules, then the molecules have no smell. Good. Just like another typical example is human being. There are people that when they pass, you perceive due to their perfume, or some people through the body odor. You must know that person. You must use something to detect. Humans have about 350 olfactory receptor subtype that work in various combinations to allow us to sense about 10,000 different odors. Compare that to mice. For example, when you have about 1,300 of linear receptor types and therefore probably sense more odors. Both odors and the tissue involves molecules that stimulate specific chemoreceptor. Although humans commonly distinguish taste as one sense and smell as another, they work together to create a receptor of flavor. In order to create it, taste. Dictating a taste gestation is fairly similar to dictate an odor or factor, given that both taste and smell lead on chemical receptors being stimulated by certain molecules. The primary organ of taste is the taste bud. The taste bud is a cluster of gastro-receptors, taste cells that are located within what? Within the bulb of the tongue is called what? Papillae. Single papillae illustration. We are going to see some illustrations. You use to taste something through your tongue. When you put something, look at the dymatical function. Look at the dymatical illustration of tongue. As I said in our previous lecture, that everything in human being seems as if it's in pairs. When you look at the dymatical structure of this tongue, you see the down, you see as if it's what is divided into the other side. When you look at it, you see phlegm papillae. In this phlegm papillae, we use it to test something. Like now, I want to test the sugar. Is this phlegm papillae that you test? If it's good for the person to swallow, the person will be able. We also swallow it. It will pass it through the esophagus. Saliva will be able to push it down. Another example, look at smell and taste in the brain. Olfactory nerve projects form an olfactory epithelium to what? To the olfactory bulb as what? As tiny. On melitated axons, the olfactory bulb is compelled and composed in narrow cluster called glomeruli, and each glomerulus receives single from one type of olfactory receive it from one type of olfactory. This is talking about taste and the smell. It's need to know, you'll be able to detect whether that one is good or not. The last structure that olfactory single directed travel to a corneal sector in the temporal lobe structure important in Spitzer autobiographical autobiographical, and others. When you look at this one, see the one that's called receptor. In that aspect of receptor, see, receptor is talking about sound. All mammal sound waves are collected by what? By external cartilaginous part of the ear called the pinna. Then travel through the what? Auditory canal and the what? Caused by vibration of the thinner different called the tympanoid in the eardrum. In this aspect, the eardrum is very, very important. In this eardrum, you can't use sharp object in that. You see, because when you something affect the eardrum, it may lead to another thing. At times, the person may not be able to receive the sound of the things very well. The person may not even be able to see. Interior to the type is the middle ear. When you look at the diagram, see the things of the ear. Look at it. The ear plays an important role. Without ear, you cannot even hear. When you look at the people that are deaf, they can't hear. They can't hear because of what? Because they have problem in their eardrum. If we do not have the melons and the insu, then the vibration of the tympano will never reach the inner ear. The vibration will never reach the inner ear. Look at it diagrammatically. Look at the ear canal. Look at the ear canal. Look at the tympana. Look at the tympanic cavity. Look at round window. Look at the external tube. This tube is very, very important. If anything happens to this tube, the person will have ear sore. Look at other ones here, different types. Even the one vibral nerve, the one that will transport sound to your ear, you'll be able to receive